Thank you for the invitation to speak to you today. We're going to start with a quiz. Students only. Who can tell me how many freshwater fishes, native species are in Canada? I'm going to pick you. How about you? You look pensive.
100.
I don't know.
Who said that? Okay, we got 100. We'll take a couple of guesses. 300?
250.
Who said that? Okay, anybody else? Okay, 250 actually is the closest. It's around 200. Well, actually 197, but there's going to be a couple of newbies due to some taxonomic changes that are coming up. So you get a ride to the Freshwater Crucible, BC. That's a $10 value. That's a $10 value. Okay, we're not finished. Who can tell me, given that, you can probably guess it's lower, how many freshwater fish species are in British Columbia native? You're on, no Googling it. What do you think?
80.
80? Anybody else?
25.
Ooh. I think you gotta have a low end. Anybody else?
120.
Well, who said that? Okay, no, you're wrong. 60, about 69. Who said 80? There you go, congratulations. Another $10 value. Okay, so hopefully you'll know a little bit more about freshwater fishes in Canada and British Columbia at the end of this talk. Certainly those with the pamphlet know a little bit more. Okay, so this is going to be the talk. I'm going to talk about the diversity of freshwater fishes. Then I'm going to talk about the problems they face and why. Then I'll talk a little bit about COSEWIC and designatable units. And I'll talk about, the last part will be some of the research that we do in my lab. And I picked this example because it illustrates one aspect of the interest people have around here on free-spine sticklebacks. But also it is directly relevant to the conservation of these fish and some of the complexity that is involved in making decisions on conservation, particularly for these kinds of fish. Should be around 35, 40 minutes. It might take me a bit longer because I got to wander around here. First thing you have to know is the fish are the vertebrate champions. They are replete with diversity. There are more named species of fishes than all other vertebrates combined. And there are more fish described every month. Open up an issue of Copia or an issue of Zootaxa, and there'll be multiple species of fish that are being described for the very first time. They just found a whole bunch of new ones at 8,000 meters off the Marianas Trench. And when we think of diversity of fishes, this is typically what, and I've done an unscientific poll. Most people, when you say, what do you think, what pops in your mind when you think about diversity of fishes? They think of a coral reef. And it's myriad species, beautiful colors, all interacting on some beautiful tropical isle. Or they might think of some charismatic marine fish, like a seahorse. And I picked that two examples for obvious reasons, particularly the seahorse. However, take note, fresh waters are wellsprings of diversity. They outdo marine waters on average by a huge amount. On the left here is the total number of amount of aquatic surface area taken up by marine waters, the big blue section, and that little tiny sliver of less than 0.01% of fresh waters. On the right are the number of described species in each of those two habitats over here. About 40% of all known fish species are described from fresh waters, even though they take up a tiny, tiny portion of the total surface area of aquatic habitat on Earth. And on a per surface area, and of course it varies tremendously depending on the areas you're looking at in these areas, but on a per surface area, freshwater fishes are 41 times as diverse per unit area than our marine fishes. And if you do it by volume, and some people have actually done these crazy calculations, it's even more extreme. So freshwaters are tremendously diverse in terms of their fish faunas. So, sure, think about coral reefs and think about seahorses, but also think about, and a lot of people do know this, about the tremendous diversity we have of cichlids in freshwater lakes in the East African rift lakes. On the left, the middle, are the species flock of little darters found in the Mississippi Basin. Scores of species found in the Mississippi Basin, all beautifully colored with amazing behavioral repertoires. And on the right are a smaller species flock of whitefishes in the Great Basin lakes. Many of these occurring in the same place. And no less a person than Carl Hubbs said, possibly the freshwater fishes, of which I have studied more than 1 million specimens. He loved to tell you about how much work he did, and that all students should be doing more than him. Are peculiar in the high percentage of forms which only by arbitrary decision can be classified as subspecies or species. And that last part is very important. We'll come back to it, because it's a problem that we face when we try to characterize freshwater fishes and how we prioritize them for conservation assessment. And some people think the reason why freshwater fishes are so much more diverse than marine areas is because speciation occurs faster in freshwater habitats. So this is some work on these silversides. 3 genera of silversides. There's a marine genus, a freshwater marine genus, and a freshwater genus. And these silversides are marine fish, and they repeatedly colonized freshwater and diversified on multiple occasions. And this is work by Devin Bloom out of Nathan Lovejoy's lab at U of T. And it's just showing the number of lineages on the y-axis accumulating through time from the past to the present, with the blue being freshwater lineages of these silversides and marine fishes in the red. And you can see in this particular case, they made the argument that, they provided statistical evidence for the fact that lineages were diversifying faster in freshwater than in marine systems. And this doesn't hold for all freshwater diversification events, but it's a good example of the idea that some people think there are particular aspects of freshwater that promote diversification, and in some cases higher extinction as well, relative to marine systems. So there may be a reason why there is this sort of disproportionate diversity in freshwater. There's something about the environment that promotes diversification. Okay, a little bit about the Canadian and BC native freshwater fish fauna within that context, that general context. This is a map of North America with the major drainage basins you can't really see too well outlined in black, and it just shows an example of the well-known latitudinal gradient of species diversity that also occurs in fishes. A couple of the, and you'll see I've got 3 highlighted here, the Arctic Cascadia, where we are in the Rio Grande. 3 numbers. The first number is the number of genera in that basin. The second number is the number of species, and the third number is the percent of those species that are endemic to that area. And you can see that in general, the Arctic archipelago is an extreme example. There's very few species, and none of them are endemic. And that tends to characterize Canadian freshwater fish faunas, that we have lower degrees of endemism and lower numbers of species in further south in the range. Cascadia, which brackets includes where we are in British Columbia, is a bit bigger. It's got 11 genera, 60 species, about 25% endemism. And the Rio Grande, 134 species, about 50% of those are endemic to the Rio Grande Basin. And if you look in Canada, Hudson's Bay in the middle is a huge area. Biggest watershed in that whole map. It's got 0% endemism. It's got about 101 species. Our most diverse area is the Great Lakes in the middle with about 168. Most of that is because of its proximity to the Mississippi, which is sort of the mother fauna of freshwater fishes in North America. Now, if you look at British Columbia, or Canada, as I mentioned, we have about 197, 198 native species. The most diverse area is Ontario, with 122, and BC is a bit of an old slide, has 67 species, native freshwater species. So we're about half of the most diverse region in Canada, which is Ontario. Our generally depauperate nature of our fauna in Canada and in British Columbia is a legacy of glaciation, destruction of freshwater habitat, elimination of populations, and the vagaries of post-glacial colonization after that. And this simulation is simply showing at the height of the last Wisconsin glaciation how all the freshwater habitat was obliterated. The glacier starts to recede, providing variable recolonization routes from these much more taxonomically rich areas in the south. And the pattern and distribution of fish we have in different areas is a reflection of the dissolution of this ice sheet, and the relatively recent time, short period of time that's transpired since that massive event that happened about 20 times over the last 2 million years. We're only living in the hangover, if you will, of the very last, most recent one. But I say this is deceptive. So the relatively low number of named taxa we have is deceptive because Canada, and British Columbia, and actually much of the, pretty much all of the whole Arctic, is characterized by a lot of genetically, morphologically, and/or behaviorally distinct populations which are of uncertain taxonomic status. They may in fact be separate taxa, we just haven't got around to describing them. As I mentioned, this is very common in freshwater fishes, both in the northern, certainly across all the northern hemisphere, but also increasingly we're discovering this in the southern hemisphere as well, areas that have been impacted by glaciation. And these different populations range from morphologically and highly divergent allopatric populations, where the real sort of vexing, or the real problems of deciding whether it's a biological species or not are most apparent, to undescribed sympatric biological species. These are sympatric populations that are behaving as distinct biological species, but we haven't figured out a way to name them yet. And as Robert Behnke said back in 1972, this is where evolutionary reality, the proliferation of these genetically distinct types that are behaving as distinct species, does not meet the sort of where evolutionary reality and taxonomy do not meet. We don't have a system to name all these divergent forms that many people believe are actually distinct biological species. So this raises a problem for conservation of this diversity, if you believe it's important, because it's relatively easier to get buy-in for conservation of something that has a distinct name, as to a situation where someone might say, oh, well, it's just a divergent population of a very widespread taxon. And there are many examples of this phenomenon, these repeated occurrences of divergent populations occurring in various degrees of sympatry, allopatry, or parapatry. And these are just several types of species. Often they're called species complexes that have these forms that we're unsure whether or not they might actually represent more than one species, more than one taxon. And of course, the three-spine stickleback at the top that is so famous here at UBC and worldwide now, which I'll spend a little bit of time on later. But I also wanted to point out that this phenomenon occurs at the very base of the vertebrate tree. So the lampreys split off from the jawfishes deep in evolutionary time. And you get the same phenomenon, divergent populations occurring in sympatry that have distinct ecology and are genetically distinct, occurring in this very basal lineage of the vertebrate tree. The same, which I find amazing, the same things are happening in these lineages of lampreys that are derivatives of that very first split in the vertebrate tree. So here are some examples, more or less local examples of this phenomenon that's proved so challenging for taxonomy. On the far left are the lake whitefish species complex, Coregonus lucaeiformis. The top are the well-known dwarf and normal types. Typically occur more in eastern Canada, throughout Quebec, but also in adjacent places in Maine, some in New Hampshire. And the bottom is the so-called humpback and normal whitefish that occurs in Yukon, the Yukon River Basin. And this is one of the, a couple of lakes in the Yukon River Basin. And this is one of the examples where we're going to get, looks like a new taxon in British Columbia. So, well, they will also occur in British Columbia, but in Canada. So these are going to be split up into Coregonus lucaeiformis and Coregonus laberatus. It looks like the European whitefish has come across Thuringia and colonized parts of Alaska and Yukon and northern British Columbia. The middle one are sockeye salmon and kokanee. Those are both 4 years. All those fish there, their males and females, are 4 years of age. They share the same lake environment for at least a year of their life history before the sockeye move off to sea. When they come back, when all these fish are mature, they're spawning in the same streams at the same time, in very close proximity to each other, a couple feet, a couple of meters apart, and they maintain genetic distinction despite some gene flow between them. And of course, in the far right are the myriad forms of the three-spine stickleback, all linked under this name Gastrosphericuliatus. There are the benthic species pairs that I'll talk about. There's stream lake pairs, there's a famous white stickleback, there's a giant stickleback, all this incredible diversity, some of which has been strongly argue that these are distinct biological species, but they all reside under this name. So how do you conserve something with all that diversity under a single taxonomic name? People tend to be fixated on it's only different if it's got a different name. Fortunately, we do have a solution. So SARA, or the Species at Risk Act, is the federal legislation that came in for protecting species at risk of extinction in Canada, which came into play in 2003. So this cryptic diversity that I'm talking about, these distinct morphological and genetic types, all residing within a single taxonomic name, is accounted for in the Species at Risk Act under this definition, this term of a wildlife species. So a wildlife species is not, it can include a taxonomic species, but doesn't have to include a taxonomic species. A wildlife species is a species, subspecies, variety for the plant types, or geographically or genetically distinct population or populations. And these are, under SARA, under the law, they are species just as much as two things that have distinct names. And at COSEWIC, we lump all of these, so species, subspecies, varieties, or geographically distinct populations, under the term designatable units, because that's the thing you designate, that's the thing you give a conservation rating for. And these are short form DUs. So DUs are defined and assessed by the Committee on the Status of Endangered Wildlife in Canada, COSEWIC, which George mentioned I'm the chair of until September of 2018. So how are DUs identified? Well, as I mentioned, they can be recognized species or subspecies, which is easy, or a variety, and varieties often have names. Or, as I mentioned, they can be a single population or an assemblage of populations, but if the latter two, if you want to carve off a single population or group of populations from the taxon as a whole, that putative DU has to satisfy two criteria. It has to be discrete from all the other representatives of that taxon, in at least one of these characteristics. It has to be genetically distinct, by any way you can demonstrate that, molecular or otherwise. Biogeographically distinct, so in a different ecozone, a different watershed, or a large range structure, east of the Rockies, west of the Rockies, that sort of thing. So they have to be discrete from all the other ones in the taxon, but, or and, that discreteness must be evolutionarily significant. It must mean something to the evolutionary legacy or the persistence of the animal or its evolutionary potential in the environment in which it lives, or plant. And significance is, that criterion is met by at least meeting at least one of these four sort of sub-criteria. There has to be something about its phylogeographic history. Did this assemblage you want to conserve exist and was isolated in a distinct glacial refuge, and therefore does it from other ones, and does it show signals of that? So is this marking part of the distinct evolutionary legacy of that lineage? Does it have local adaptations that are not found, as the name implies, anywhere else, that are important to the legacy of that, the past and the future evolutionary potential of that species to exist across the landscape? Would the loss of that assemblage of populations induce a large disjunction in the range? Or is it the only remaining natural occurrence of that taxon? It may, the populations may exist in other areas, but they're outside their native range. And is this the last remaining representative in its natural range? So here are some examples. The caribou is, of course, always in the news. Rangifer tarandus. There's 12 DUs in total. 2 of those are recognized simply on subspecies status. There were a lot of other subspecies, but we abandoned those because they didn't make any sense. But the Perry caribou and Dawson's caribou, the latter one is extinct, are 2 of 12 designatable units of caribou. The lake sturgeon, Acipenser fluviescens, is found in 4 DUs grouped by major watershed. And the American badger, here's an example. We have 1 subspecies in Canada, but that subspecies has now been split into 2 DUs, eastern and western designatable units, because they satisfy one or more of those criteria that I talked about before. So this provides a rational way in which to recognize diversity below the taxonomic level that people feel is important to conserve, both as a tribute to the evolutionary legacy of these things, but also to safeguard their evolutionary potential. That's the thinking anyway. And this is important for freshwater fishes because there are a lot of designatable units in freshwater fishes. This shows that on the y-axis is the total number of wildlife species assessed by COSEWIC. in freshwater fishes and marine fishes. So we've done about 150 freshwater fishes. The dark blue represents the number of the light blue that are designatable units within a taxon. So you can see half of the wildlife species that we assess consist of population assemblies, single populations or population assemblies within a single taxon. In marine fishes, you can see that about the same pattern. We've done fewer marine fishes, but even a higher proportion of those are designatable units within a taxon. And the very last column shows the number of the dark blue that are represented by anadromous salmon. So marine fish group does anadromous salmon for reasons that I won't get into. But there's a lot of designatable units in salmon. There's quite a few in Atlantic cod, but most of this surge in designatable units in marine fish is because of marine fish. So for instance, there's 29 designated units of sockeye salmon in the Fraser River alone. Just in the Fraser River alone. Those are all species under CERA. Okay, so let's move to the next part, just to talk about, we've got some handle on diversity of freshwater fishes, what the Canadian situation looks like, and now I want to look at the state of freshwater fishes, or conservation state. Generally, the status can be classified as poor. And this is part of a general trend of declines in numerous freshwater taxa. In North America, it's been estimated we're losing about 4% decline in named or accessible units per decade, which has been suggested to be 5 times higher than for terrestrial taxa. In Canada, we've had 9 extinctions and extirpations of freshwater fishes, and 94 wildlife species have been assessed at some level of risk by COSEWIC since it started in 1978, since we started assessing things in 1978. And that's anything from special concern through to extirpated, including endangered and threatened. And that's about 77% of the total we've assessed have been found at some level of risk. This is part of a global phenomenon. This is the top panel shows the number of, on the left shows the number of described species per million square kilometers. That's shown in the bars. And the lines show the number of IUCN, International Union for the Conservation of Nature, imperiled species per million square kilometers. And this is for all freshwater taxa, not just fishes. And you can see the problem is immense in freshwater habitats. The panel shows, the lower one just shows for the US, these are birds and mammals. These are all 3 different types of freshwater taxa. Everything but the white bar represents things that are of least concern or secure, and all the dark shaded ones represent things that are of some level of risk. And you can see the proportion of the total pie is much higher for these freshwater taxa than for terrestrial taxa. So it's sort of widely acknowledged that freshwaters are in real trouble. This is the numbers in Canada that have been assessed at some level of risk, special concern, threatened, endangered, or extirpated. This is extinct, extirpated, endangered, threatened, special concern for marine and freshwater, and you can see that freshwater fish tend to have a higher risk status than marine fish. This is going to change big time when all the salmon, if they do in fact get listed, because that's going to really increase the marine fish group. But in terms of extinctions and extirpations, as I mentioned before, this has only happened in freshwater systems. The geography of species at risk in Canada. The bars, this is a bit dated, but the height of the bar shows the total numbers of species that have been assessed at some level of risk in Canada. And the places with the highest bars tend to be southern British Columbia, the prairies, and southern Ontario. That goes for the total number of assessed, and the total number that are actually end up being listed, which tends to be a much smaller fraction, by the federal government. So we assess things, it's the federal government that actually lists them. Why are freshwater fishes in so much trouble? Well, it's essentially because they're entombed within a terrestrial landscape. Anything that happens in the terrestrial landscape around the waters, around the river or lake, is going to eventually end up in the lake or impacting the lake. There are multiple stressors, not only going directly into the water, but also on the landscape that filter down eventually into the watersheds. There's more than 1 million dams worldwide already. And we capture about 50% of the surface flow already, which has tremendous impacts on the biodiversity of fishes and other freshwater things. Habitat loss, degradation, invasive species, water security issues, etc. The very bottom ones are legislative changes that weakened environmental legislation that the Harper government prosecuted, and those are in the process of being changed, hopefully for the better, by the Liberal government. This again is a little data from 2006, but it shows for COSEWIC assessments the threat factor that is most important for at-risk status for freshwater fishes. Number one, either the primary factor or the secondary factor is habitat alteration, destruction, or loss. And if you add pollution into that, which I would, it's obviously even much higher. And alien species typically tend to be the second most important factor that is implicated in the threat status of freshwater fishing. Now, that's a dated slide, but this is a very recent example of this issue. So the Port of Montreal wants to expand their port eastward into critical habitat for species at risk in Canada. And these include two aquatic or semi-aquatic species. One, the western chorus frog, and more importantly on the right, an endemic species. Canada has 100% global responsibility for the survival of the copper red horse on planet Earth. And this port expansion wants to encroach on that habitat. So even just a few weeks ago, these pressures are constantly in the mix. Now I wanted to go back to this because I wanted to read you a part that's in the box. So not only is development on land base and its impacts on either directly or directly on the watershed are an issue, but also the attitude that some people have towards development makes it even harder to deal with these things. So the quote says, port officials insisted, they didn't suggest, they didn't hope, they didn't wish, they insisted the possibility of a fish and amphibian derailing the project are remote. This is a Canadian endemic, found nowhere else in the world. And these people think the chances of that fact stopping this project are remote. They said mitigation measures would be implemented, including recreating a habitat for the red horse downstream, where conditions are of higher quality. Even though the fish doesn't live there currently, somehow they think it's of higher quality. And the other thing that amazes me, and maybe just bad reporting, the other thing that amazes me about this kind of wording is that they're so absolute. They're certain that they can fix the problem. Time and time again, that's proved not to be true, and this is another problem that freshwater habitats face. Okay, so that's sort of the general background, and the goals of sort of my COSEWIC-Inspired work, which forms part of what I do in my lab, is essentially to test the veracity of DUs in several wildlife species. So I've got several listed here that we've all worked on, from Nooksack dace to Lake Chub. Many of these single taxa have multiple designatable units. Many of those multiple designatable units were erected based on, in some cases, anecdotal information, in some cases just morphological variation, in some cases just based on geography. And there wasn't a lot of sort of hypothesis testing or rigor in actually assessing whether these things that are going to command resources and are going to change people's lives actually merit being on Canada's species at risk list. The second part of the things we do related to COSEWIC work are genetic contributions to threats assessment and monitoring. So we've done a lot of work on the issue of hybridization between introduced rainbow trout and native western cutthroat trout, and on the stickleback species pairs, which I'm going to focus on for the last bit. Okay, so Enos, lake, benthic, and limnetic three-spine sticklebacks, endangered or extinct. And I'll explain what that means in a minute. The top is a benthic male, and the bottom is a limnetic male from Enos Lake on southeastern Vancouver Island. Okay, for those of you who don't know anything about sticklebacks, which I can't believe, I've got benthic male and female on the bottom, limnetic male and female at the top. This is from Paxton Lake, I believe, maybe Priest Lake, I can't remember. So these fish are about that long, and they are morphologically, ecologically, and genetically distinct in sympatry. They occur on 3 islands. Well, they used to occur on 4 islands, 1, 2, Texada, and Nelson Island in southwestern British Columbia, and here's just typical small little coastal lake. These things are behaving as biological species in sympatry, and they are arguably some of the youngest vertebrate species on Earth. And they're called benthic lunetics because the benthics tend to spend most of their life, they spend their entire life in the littoral zone, where they feed, grow to maturity, and reproduce amongst aquatic vegetation. The lunetics spend much of their life out in the open water in schools, feeding on plankton, and then they come inshore, mate next to the benthics, but in slightly different habitat, and the two do not interbreed, mostly in nature. So they maintain themselves as behavioral and genetically distinct units in sympatry, meeting the definition of what most people think are biological species. And the plot in the middle just shows gill raker count. So these things feed on different things. The benthics have lower numbers of gill rakers, tend to feed on benthos. The lunetics have a higher number of gill rakers, tend to feed on plankton. These are very, very well established for decades. These things have been these sort of stable entities. And they are, as I mentioned, they're found in southwestern British Columbia in the sort of post-glacial submergence zone. So this is Vancouver Island. The gray is the post-glacial submergence zone. And the blue lakes are on Texada and Nelson Island, where there are extant populations. And the two red ones are historical populations that have gone extinct on, we'll see if they've gone extinct, on Enos Lake and Beaty, Pete's Lake or Hadley Lake on Beaty Island in the middle. So there was historical occurrence in 7 lakes, I think. One pair has gone extinct in Hadley Lake because someone introduced a catfish and ate them all, basically. And these are all SARA listed as extinct, endangered, or threatened. So what that means is there are prohibitions against harming them or killing them or destroying their habitat, and there has to be a recovery plan. So resources have to go into recovering these things to some predetermined level. So in the early 2000s, people from my lab and people from Dolph Lab collaborated on a project to try and assess, there was some indication that these things hybridize naturally in nature, but despite this hybridization, they maintain themselves as distinct entities. But there was some indication that the level of hybridization in Enos Lake was increasing for some reason over historical times. So we presented evidence in favor of this idea that speciation had gone in reverse and created a hybrid swarm between these two distinct entities that were now a single pile. And we did this using historical morphological data. So this shows a time series of morphological data from 1977 to 2002, just a summary score of morphological variables. You can see in 77, when they were sort of first discovered, very clear distinction morphological between them. 1988, it's still there, but it seems to be collapsing. By 1997, it had gone kaput into a single mush. These two very distinct things appear to be a single morphological group. We also showed this genetically. So for comparison, there's the Priest and Paxton Lake pairs represented by those two distinct, very distinct genetic clouds of points. And by 1994, the Enos Lake group appeared as a single sort of genetic mush, which was consistent with morphological data. Unfortunately, we didn't have historical data for the genetics of these fish, although historical Alazine data clearly showed that they were very different genetically. It probably happened because of the introduction of an exotic crayfish. We're not exactly sure why this happened, either direct predation, particularly on one type versus the other. Jordan and I had certainly habitat disturbance. These things are omnivores and they just eat everything, including aquatic vegetation. It's important for breeding. And Jordan and I had a student, Garrett Belima, who looked at the nesting behavior of males of benthic lunetics in the presence of crayfish, and he showed that the crayfish seemed to have, the lunetic males seem to be very timid, and this may have impacted their breeding behavior in the wild, which through some unknown mechanism may have facilitated hybridization. Okay, so what's this thing about endangered versus extinct? Well, we presented evidence, morphological and genetic, for the presence of a hybrid swarm. We did this, and so did other people who were looking at nuptial coloration, Malik et al. So what are the implications for this collapse of two things into a single cloud for listing and recovery? In other words, should they be delisted? Each one was a separate listed species as endangered. Now they've collapsed into a hybrid swarm. Should they be delisted? Should recovery actions, which means recovery potential and going out and maybe getting rid of the crayfish, replanting aquatic vegetation, doing a captive breeding program, which all costs money and time, should that proceed? Well, I argued that it shouldn't. I argued that these things were, at that time I was a co-chair of the freshwater fishes, I argued that they should be declared extinct because of this sort of genomic extinction that we demonstrated. And people are very, and rightfully so, are wary of declaring something extinct, because that's basically like accepting the null hypothesis, saying there's none left anywhere. And the question came up, was the sampling sufficient to declare limetics and benthics as extinct? You showed the populations collapsed, but how do you know there isn't one or two lonely limetics and benthics swimming around the lake that you could capture and bring into a captive breeding program and all this sort of stuff? Because extinction means no individual exists anymore, or no reproductively competent individual exists anymore. So we sought out to address this limitation, that all the studies that we've done before where we quoted levels of hybridization were really parts of other studies. They weren't specifically designed to look at hybridization. The problem was we didn't have historical data. But the reasoning I came up with was, well, we have these 3 other intact species pair lakes. Let's compare the genetic situation of those lakes, where we know there are 2 discrete types, to the situation in Enos Lake. Can we use these as a standard, in the absence of historical data, to come up with a sort of firmer, more robust conclusion about whether any single individual limetic or benthic exists? So we sampled the lake extensively over 2 years. The yellow lines are the transects. Over 1,200 fish were sampled across 3 different seasons, different life history stages across 2 years, setting traps in the bottom, setting traps in the littoral zone, setting traps suspended from buoys to try and sample the limetic zone. And the gill rakers, to me, really say it all. These are gill raker counts that McPhail, from samples McPhail collected in 1977. We recounted them just to make sure everything was okay. 2 very distinct modes, benthic on the right with about 18, limetic on the right with about 24 average. This is just one of the samples of the 1,200, smaller group of samples. Clearly a unimodal distribution with about 21 gill rakers on average. Interestingly, when you cross these fish, make artificial hybrids in the lab, the hybrids have about 21 gill rakers. Exactly what we find in the field, which is consistent, it doesn't prove it, but consistent with the smush being hybrids and not some other sort of new type. Now, sort of to finish off, I'll just show you the genetic data we've collected. This is an output from Structure, which is a sort of a model-based clustering algorithm that basically you give it a bunch of multi-locus, multi-allelic data, and you say, calculate the relative likelihoods of some number of populations, genetically distinct populations that best describe these data, where the populations are obviously 1 up to some number. And along the bottom are the numbers of fish in each case. So each fish is represented by a very thin vertical line, and the color composition of that line represents the relative proportion of 1 of 2 genetic groups characterizing that one individual based on these microsatellite loci, with 1 genetic group being green and 1 genetic group being red. Right, so when you do this, the most likely number of populations in these intact species pair lakes is always 2. Not 3, not 4, not 1. It always says there are 2 genetic groups, 1 green and 1 red. And the Q value on the side goes from 0 to 1. This represents sort of a summary score of the genetic composition. So a fish with a Q value of 1 is 100% green. And it'll have a Q value of 0 if you did it in terms of the red one. So these red fish over here have a Q value of 0 because they're 100% red, not green. So it gives you can assign an individual fish after you decide how many populations there are. You can assign individual fish to one of those 2 clusters or a mixture of those 2 clusters. So you'll notice that most of these fish are all green or all red. There's a few that are admixed. This is a red one that's got 40% green of the green sort of genetic group within it. Now what I did after was I mapped onto these genetic groups what these fish have been characterized as morphologically from the gill rakers and some other measures. And you can see there's almost perfect correspondence. The greens are limetics and the reds are benthics. Now, if you look at Enos Lake in 1994, you see a slightly different situation. Yeah, there are 2 genetic groups, although the most likely solution here was actually just 1. But what I did is I forced 2 groups on the data and said, if I assume there's 2 groups here, how do they map onto these 2 groups? And again, most of the benthics are green, most of the limetics are red. You can see there's a much greater level of uncertainty about the genetic composition of any individual fish. There are no fish that are 100% red and no fish that are 100% green. And this just states that. There you can see in the top, there are no fish with a Q value greater than 0.99, or, that is the green population, or none with a Q value of less than 0.01, which represents the red group. Whereas the lower three panel, these three intact species pair lakes, virtually all of them are 100% one or the other. There's some mixture between them, and we knew that before. So the idea was to use these as a standard to evaluate whether there are any pure individuals in Enos Lake or not. Now, the situation gets a lot worse when you look at over 1,000 fish in 2013-2014. You can see it's total admixture. Most fish are about half one and about half in the other. Totally different than this, and it's gotten worse since 1994. The sections between the dark black lines in the second from the top, those represent fish that were caught in linetic traps, the traps suspended in the water column. And there was no, the linetic fish were no more red or green than the other fish. And this, if you plot the admixture scores, so this is again, under a hybrid would be 0.5, an F1 hybrid would be 0.51, the red group, and 0.5 the green group. So these are the 4 sample periods we did. These are the admixture scores. You can see clearly there's a single unimodal genetic population of fish in this lake, no matter what season you sample in, where you sample in the lake, whether you sample juveniles or adults. Now look at, this is a situation for Priest Lake, where we have these intact fish, intact species pairs, as shown in a different way. You can see by far most of the fish are either strongly one type or strongly the other type. So this situation has, Priest and the other 2 lakes, has morphed into this situation. And we think it's because of this variation. So, to conclude this part, and basically the talk as well, as distinct populations, Enos Lake benthic limetics no longer exist. I think we've shown that very extensively. They've collapsed into a hybrid swarm. The unimodal gillraker counts and genetic Q scores that I just showed in the last slide. More importantly, there's no fish with a Q value greater than 0.99 or less than 0.01. And so they do not meet in Enos Lake. They do not meet the standard of, shown in 3 independent lakes, of Q values greater than 0.99 or less than 0.01 that are strongly tied to the phenotype that we care about, that is whether they're benthic or limetic. Now, even if one of those species did exist, so let's say you had a green population, say you show there are green fish of Q value greater than 0.99 that represent benthics, I would argue that even if one of the species exists in the lakes, their significance is no longer significant. Because the significant thing about Enos Lake, or all these pairs, is these 2 things occurred together. And their interactions together were important in the past, and they're important currently to maintaining, or not, their distinction as 2 biological species. So even if you had one but not the other, I would argue that lake has lost its significance in terms of conservation. Not everybody agrees with that. So this, all this to say that in my view, the work we've done supports the extinct genomic status, because there's still hundreds, if not thousands, of fish in the lake. Just it's the unique combinations of genetics and morphology that define historically benthic limetics that no longer exist in that lake. So this is where things get tricky, because first of all, people can't get their head around the fact that, well, you say they're extinct, but there's lots of fish around in the lake. That's right, but they don't have those unique combinations that I talked about. So you could get people's head around, okay, well, let's talk, then they're genomically extinct. But then there was the issue, was the sampling extensive enough? And can you actually say there are no individuals left in that lake? And I think, unless someone wants to put even more money into this and sample them even more, and you could argue you can sample them more extensively from the genome in terms of whole genome sequencing, I would argue this supports a reassessment of these fish as extinct. So, to conclude, freshwater fishes are amazing. There's lots of unappreciated diversity in society in general in freshwater. Canada has a relatively low diversity of formally described species, but we have this tremendous cryptic diversity that is a function of our geography, which I think is really cool. Geography and diversity tied together explicitly. COSEWIC-Inspired provide a mechanism to document, assess, and protect such diversity. That's the DU concept. However, a lot of this stuff falls apart because they don't get listed, and they don't, even if they do, they don't get enough recovery action because there just aren't the resources, and sometimes there's not the political will to just get on with it, which is what we need to do. So thanks very much. I think that